So let's focus a little bit on proteomics, which is the identification, characterization, and analysis of all proteins encoded by the genome of a cell. One of the things you can do with this is try to reconcile differences between the number of genes in an organism and the number of proteins that are produced. You can also compare proteins that are found in normal tissue compared to diseased tissue. One of the techniques you can do for this is called 2D Gel Electrophoresis. It's a technique for separating hundreds to thousands of proteins with a high resolution. So what you do, you've got your cells or tissue that you're growing in vitro. You give some of them condition A, some of them condition B, one of them might be a control, one of them might be an experimental variable. You'll grow these cells up, you'll extract the proteins, you run them on a 2D gel. This gel will separate them out not only based on size, but also based on pH. So here's a charge here and a charge here. What you look for are for spots that are different between these two gels. So you can see most of these spots are the same, but hey, look at this spot right here. It's present here, but not present here. So this is something that's changing when you're changing this condition. You can cut out that spot and you can go through and do mass spec and you can compare to protein databases to try to identify what was that protein in that spot. Okay, so let's talk about this, how this is working, this 2D gel stuff. So the first dimension, you're going to load your protein sample onto this focusing tube gel. This is going to separate out proteins based on their pH, their isoelectric point. So their net charge is 0 compared to the pH of the gel. Okay. Then you run them in a second dimension. So you have this gel on here, and then you place a charge on it. So this is going to be separating these proteins based on their weight. So smaller proteins will go quickly, the larger proteins will be stuck in this gel. And this is what a sample like this might look like. Here's another example. Here's a 2D gel separation of human platelet proteins. You can see that many of these have been identified. Many more of them have not yet been identified. This mass spec, so what you can do is you can literally cut that spot out of the gel and you can run mass spec on it. What you can do for this is you can literally determine what is the amino acid sequence of that particular protein. You can at least get the start of a peptide, and you can use that to compare to protein databases to try to identify that protein. Oops. So here, here's a 2D gel, here's an unknown spot. Okay, here's our protein. You're going to cut that out. You digest it into little peptide fragments, and you subject these to the mass spec, and it'll give you a particular spectra. You can use this using a database, you can compare it on the computer, and what it'll do is it'll give you a sequence. In this case, this is the particular sequence that it would be here. You can then try to map that peptide to other known proteins that happen to have that same sequence. Okay. Here's a cool example. They did mass spec analysis of bone tissue from T-Rex. It's estimated over 68 million years old. So what this demonstrated was that this fossilization did not fully destroy all protein if it was a well-preserved fossil. So here's the experimental peptide. Here's a synthetic version of the peptide. And this is what they think the sequence is for that particular region.